in today's lab, we're going to differentiate using different reagents between main biological macromolecules, carbohydrates, lipids, proteins, and nucleic acids. So these are four major types of biological macromolecules. Carbohydrates can be divided into monosaccharides and disaccharides and polysaccharides. Monosaccharides consist of one sugar, basic subunit of carbohydrates, for instance glucose. Disaccharides consist of two sugars, two monosaccharides. Sucrose, maltose, and lactose are good examples. Finally, polysaccharides contain many monosaccharides, and compounds like starch, cellulose, glycogen are some examples of these polysaccharides. Lipids are nonpolar, and that's important. Nonpolar molecules. Major groups of lipids are triglycerides, waxes, phospholipids, and steroids. Triglycerides are formed during the esterification of fatty acids by glycerol. This is why they're called triglycerides. 3 fatty acid molecules and the glycerol. Waxes are esters of very long chain alcohols and fatty acids. So beeswax would be an example. And phospholipids formed by glycerol, 2 fatty acids, phosphate group, and amino group. Phospholipids comprise the cellular membrane. Steroids are the only group of lipids that are not esters. Steroid structure consists of 4 carbon rings that are fused. They form a nucleus, and then this nucleus is modified by the side chains. So cholesterol, bile, and testosterone are examples of steroids. Proteins, very diverse. They're polymers of amino acids. They perform a wide variety of functions, from structural to signaling to facilitating the reaction as enzymes, storage, protection. So we're going to use, for instance, albumin in this lab. Nucleic acids are molecules of DNA and RNA. They consist of subunits called nucleotides. DNA is storing genetic information, while RNA assists in the expression of that genetic information to synthesize proteins. So in today's lab, we have 20 tubes with 4 different biological molecules there. So you can see them right here, 20 tubes. This first row contains corn oil. That second row contains albumin. The third row contains starch. The fourth row contains glucose. And the last final row is empty. This is going to be our control. And here you see 4 reagents that we're going to be using. Benedict Reagent is used to determine the presence of monosaccharides. Buret Reagent used to determine the presence of peptide bonds, so basically proteins. Iodine Reagent that is used to determine the presence of polysaccharides, and water to test for solubility. So we're going to start with adding 1 dropper full of water into each tube in this particular column. So what can we see? We can see that, well, the glucose here, this tube, and just water, they don't really look different. Glucose forms a true solution. Albumin, this tube, and starch, this tube, they both form colloid solutions in water. So it's not true solution, but it's pretty good colloid. And now we can look at corn oil, and you can clearly see that corn oil floats on top of water. They don't mix with each other. Okay, so obviously corn oil is not soluble in water. Next, we're going to add iodine reagent. It's a water-based reagent, so I can tell up front that it's not going to react. So corn oil, okay, nothing happens there. They don't really mix that well. They just mix with albumin. Nothing really happens here. And voila, you have a mixture here. You have a color change. Okay. No change in glucose, and no change in the control. So next we're going to use Buret reagent. I'm going to add a dropper full of burets to each tube. Again, this is the water-based solution, so you can easily imagine corn oil is not going to mix. It stays on top. And then we have albumin, and we clearly see the color change compared to the control. So this is the control, this is albumin with burets. And nothing really changes if you compare it to the control in starch and glucose. Basically, it just gets a little bit diluted. And the last one is Benedict's reagent. So now it is time for the Benedict's reagent. So basically we're going to add Benedict's reagent and then place the tubes into the water bath. We need to make sure that we can tell them apart. So this is going to be corn oil. Well, it's really hard to miss. So we're going to do corn oil and albumin. We know that there's something written on albumin tube. And then we have one here written on the starch tube. And then we have these two, so very little for the control that we know, and glucose is the next one. So we're going to take a look at the results in a second. We're going to place them back in the tube rack 1x1. So nothing happened with the corn oil. Interestingly enough, it's possible that albumin was contaminated with Buret reagent, so we're not going to count it as positive. So this is the starch, and it's interesting, you can see a little bit of a yellow on top. It's probably due to some presence of simple carbohydrates in the starch. And here you have a solution of dextrose, so simple starch, basically glucose. Okay, it turns yellow. And control, here nothing happens to it. So at the end of the day, what we've got there is the following. So our glucose solution got positive after the addition of Benedict's reagent, starch solution got positive after iodine, protein got positive after Buret's, and triglyceride, no solubility here. It controls, no changes. So you should answer these questions. It can be on your lab assessment, and that'll be it for this lab.